wish I could be there with you in person, but hopefully you guys will enjoy these virtual labs and also learn from them. So the first experiment we're going to be doing is we are testing 5 different solutions for the presence of monosaccharides. Now, monosaccharides is just a long word for simple sugars or small sugar molecules. These are the types of sugars that you would find in candy, soda, any of your sweet treats. So we are going to test for these monosaccharides by using a reagent called Benedict solution. This is our Benedict solution right here. You'll notice it's a light blue in color. And we're also going to need to use heat today. So with the Benedicts and the heat, if there are monosaccharides in our solution, we will see a color change. Specifically, if we have a low concentration of monosaccharides in our solution, we will see a yellow or green color change. If we have a high concentration of monosaccharides in our solution, we will see an orange or red color change. So let's go through all the solutions that we're going to be testing today. So our first solution, this is simply distilled water. Now, distilled water is going to act as our negative control. What that means is that there are no monosaccharides here, so it should always test negative. If for some reason we do get a color change and a positive test result, we'll know that something went wrong with our experiment and we'll need to try it again. Our second solution is glucose. Now, if you're not familiar with all these solutions, that's okay. These tests are going to help you learn what's actually in them. So hopefully by the end of the lab, you'll be more familiar. In our test tube 3, this is a substance called albumin. Test tube 4, this is starch. And test tube number 5 contains cooking oil. Now, most of these test tubes, the solution they contain is transparent in color. However, test tubes 3 and 5 do have a yellowish tint. Now, that's important because when we add the blue Benedict solution, they will most likely turn to a greenish tint. Now, that color change doesn't necessarily indicate monosaccharides. That's simply a result of adding a yellow solution to a blue solution. So let's go ahead and do the first step of this test, which is adding the Benedict solution. And we're going to add about 20 drops of Benedict solution to each test tube. So let's go ahead and do that now. Now that we've got our Benedicts added in, let's take a look at the colors. So test tubes 1, 2, and 4 really all share the same color. And that's going to be a light blue. And again, that's simply resulting from the original color of our Benedict solution. Now test tubes 3 and 5, like I mentioned, have turned to be a greenish yellowish tint. Again, that's because their original solutions were yellowish in color, and we added blue Benedict solution to them. Again, we need to use heat in order to really see if there are any monosaccharides present here. So I'm going to add these test tubes to our hot water bath. We're going to let them sit for a couple minutes and see if there are any color changes. I've been in the hot water bath for a few minutes. Let's take them out and look and see if there were any color changes. So let's start with test tube number 1 here. So as you can see, we really didn't have any color change. We still have that light blue color that's again from the Benedict solution. Let's take a look at test tube number 2. Now test tube number 2, we have a nice orange color. So if you remember, orange was one of the colors that indicates a high concentration of simple sugars or monosaccharides. So that's going to be a positive result. Let's take a look at test tube number 3. Now we did see a color change with test tube number 3. We have almost a purple or grayish color here. However, that's not one of the colors that gives us a positive result for the monosaccharide test. Again, what we're looking for is a color change to yellow-green, which would be a low concentration of simple sugars, or orange or red, which would be a high concentration of simple sugars. We're not seeing that here, so that is a negative result for our monosaccharides. Let's take a look at test tube number 4. So again, we have our light blue color. That's from our Benedicts. No color change here, so that's going to be a negative result for simple sugars. And lastly, test tube number 5. Now, if you remember, test tube number 5 contained our cooking oil, which was yellow, and again, we added our Benedict's, which is blue. So a yellow or green color is not indicative of monosaccharides here, simply a result of adding yellow and blue solutions together. So our 5th test tube is also a negative result for monosaccharides. So you're going to want to take a look at your handout and record all of the results from this experiment. So record the initial colors that we saw in the test tubes, record the colors that they have changed to, which we just went over, and record whether or not that means it's a positive or negative result for monosaccharides or simple sugars. So you can go ahead and do that now.